Welcome to today's ASBMB transcription webinars. The monthly transcription webinars aim to promote collaboration and knowledge sharing among scientists interested in the general areas of transcription, chromatin, and epigenetics. Today we have two outstanding speakers. Each speaker will talk for about 20-25 minutes, followed by answering questions for 5 minutes. After both talks, I will open 2 breakout rooms, one for each speaker to answer additional questions for 10 to 20 minutes. Today, Dr. Jessie Zhang from UT Austin will co-host with me. So when the audience asks questions, please raise your hand. You may also ask questions in the chat box. So our first speaker is Professor Bachir Affar from University of Montreal. The title of his talk is What the BAP1 deubiquitinase must overcome to reach its destination and regulate the epigenome. Welcome, Bachir.
Thank you. Do you hear me?
Yes, we can hear you.
Share my screen.
Yeah, we can see your screen.
Okay, great. Well, thank you, Kay, and thank you, JC, for the invitation. It's really a pleasure for me to join this webinar and to talk about our work on BAP1. And I will basically try to summarize almost 20 years of work in 20 minutes. And I hope I will, there will be some take-home message at the end. So my lab is interested in ubiquitin signaling in the context of chromatin biology, in the context of transcription and DNA repair, and we are mostly interested in the reverse reaction of ubiquitination, which is deubiquitination. And this reaction is actually very regulated. And we have a large family of enzymes. Most of them are cysteine proteases, more than 100 enzymes that are cysteine deubiquitinases. And there is also a small family called the JAM family, which are metalloproteases. They use zinc for catalysis. So we have been asking these questions on deubiquitinases, which are basically how these enzymes coordinate chromatin-associated processes, how they are regulated, and whether there are defects in deubiquitination that can promote human disease, in particular cancer, and whether we can learn something from that to help design some diagnostic and some treatment strategies. And the protein that I will be presenting today is BAP1, which is also termed BRCA1 associated protein. One has been discovered by Frank Roscher almost 30 years ago. It has a catalytic domain in the C-terminus, in the N-terminus, my apologies, and it has long C-terminal extension with this C-terminal domain and motifs for interaction with other proteins and nuclear localization signal in the C-terminus. So it is widely expressed, and we know from many studies that BAP1 overexpression or depletion impact cell proliferation, and that the BAP1 knockout is embryonic lethal. So BAP1 is frequently mutated and inactivated in cancer. It's a tumor suppressor gene, although we have some evidence that BAP1 in some contexts can also act as an oncogene. So BAP1 is frequently mutated in mesothelioma, in melanoma, in liver cancer, and in renal cancer as well. And we know that we can lose both copies of BAP1. So it's an inactivation of BAP1. So BAP1 is definitely a classical tumor suppressor gene, although, as I mentioned, it can also have some oncogenic properties. So the questions that we have been asking is how BAP1 actually acts as a tumor suppressor gene. And this purification actually has been done by myself almost, I will say, 20 years ago. Basically, we generated cells stably expressing BAP1, and we prepared nuclear extract, and we did a purification with FLAG and HA, and we did mass spectrometry of the BAP1 associated proteins. And at that time, we had a hypothesis. So that, again, it's more than 20 years ago, is that BAP1 is a chromatin, at least the mammalian BAP1 is a chromatin regulator. And we have been working on this BAP1 interacting proteins and complexes and defining how they interact together and how they regulate the epigenome. And in the same time, Jörg Miller published a beautiful Nature paper in which he reported that BAP1 in Drosophila is called Calypso, interacts with this protein ASX, additional sex comp, to form what he termed the polycomp repressive dub complex, the deubiquitinate histone H2A on lysine 119 or 118 in Drosophila. And the BAP1 complex is basically the ortholog of the PR dub complex in mammalian. And although there are few deubiquitinases that have been reported to deubiquitinate histone H2A, we believe that BAP1 is a major one. We did this RNAi screen, at least in these U2S cells, detecting the increase of H2A hub on lysine 119 with a specific antibody, and BAP1 stands as a major hub for this modification. So today I will be telling you 3 small stories. First on how BAP1, which is synthesized in the cytoplasm, of course, gets more or less the permission to enter the nucleus. There is a quality control mechanism that will allow BAP1 to enter the nucleus. In the nucleus, BAP1 interacts with these factors, ASXs, that basically regulates its catalytic activity. And the third part will be on BAP1 being recruited to chromatin to ensure its function as an epigenetic regulator. So in respect to its interaction, in respect to the quality control that basically permits BAP1 nuclear entry, it's in respect to its interaction with this protein called UBE2O. And UBE2O is an E2 E3 hybrid. So this is a large E2 with highly conserved domains that we termed conserved region 1, conserved region 2, and C-terminal region, and this is the UBC, the catalytic domain. And we found UBE2O as a substoichiometric component of the BAP1 complex. And we asked the question, what is the relationship between UBE2O and BAP1? And to make a long story short, we found that UBE2O selectively ubiquitinates BAP1 in the complex. So BAP1 is the only protein in its complex that gets ubiquitinated directly by UBE2O. And what is interesting is that the catalytic dead BAP1 is a better substrate than the wild-type BAP1 for UBE2O. So this is to keep in mind. I will come to it later. But first, we wanted to know what is the nature of this ubiquitination of BAP1, whether it is polyubiquitination or multi-mono-ubiquitination, because we can see many bands on BAP1. So basically, what we used is either a methylated ubiquitin that cannot form a polyubiquitin chain, or an ubiquitin which is mutated on all lysine residue that cannot form, again, polyubiquitin chains. And what we found is that by using methylated ubiquitin, we can see that we have almost the same pattern as the wild-type ubiquitin in respect to this ubiquitination of BAP1 by UBE2O. And also, when we express a mutant of ubiquitin that cannot form polymer chains, we have exactly the same pattern of ubiquitination between the wild type and the mutant. This indicating that UBE2O multi-monoubiquitinate BAP1. We mapped the site of ubiquitination, and it's the nuclear localization signal that gets monoubiquitinated by UBE2O. So this is the site for interaction and ubiquitination by UBE2O. And I will not go into details for the mappings. This has been published, I will say, a long time ago. And to make a long story short, what we found is that by multi-ubiquitinating BAP1 on its NRS, UBE2O promotes the cytoplasmic localization of BAP1, and this is the catalytic dead UBE2O. And again, as I mentioned in the beginning, the catalytic dead BAP1 is a better substrate for UBE2O. So therefore, UBE2O multi-monoubiquitinate BAP1 on its NRS and promotes its sequestration in the cytoplasm. So we wanted to understand why the catalytic dead BAP1 is a better substrate, and we had this hypothesis at that time that BAP1 fold on itself, and bringing the NRS of BAP1 in close proximity to the UCH, the catalytic domain of BAP1, through this interaction between the colloid coil motifs in BAP1. So basically, what we did is first to make sure that, in fact, the catalytic dead BAP1 is a better substrate for ubiquitination. And when we inactivate in vitro BAP1 with NEM, which is a reagent that will alkylate and inactivate the cysteine, the catalytic cysteine of BAP1, we can see nicely that BAP1 become, again, a better substrate for UBE2O. So we did a lot of mutations to disrupt this interaction between the CC1 and the CC2. And again, to make a long story short, this is essential for basically having BAP1 protected from ubiquitination by UBE2O. And here we see the ubiquitination. When we inactivate these domains, we have an increased ubiquitination and an increased cytoplasmic localization of BAP1. So basically what we have is that BAP1, by properly folding on itself, can self-deubiquitinate, can auto-deubiquitinate. And if we have that proper folding and we have that self-deubiquitination, BAP1 resists this ubiquitination by UBE2O and can finally enter the nucleus. So we have been also lucky to identify mutants in melanoma. So these are small deletions or insertions in the colloid coil domains of BAP1, and these mutants basically promote the cytoplasmic sequestration of BAP1 because we don't have these intramolecular interactions that promote the self-deubiquitination. So the take-home message is that BAP1 has to be properly folded to be able to self-deubiquitinate, and that self-deubiquitination is a guarantee, if you want, that BAP1 can go into the nucleus. So in the nucleus, BAP1 is properly folded, but cannot act as deubiquitinase for histone H2A lysine 119. And this has to be ensured by the ASXL factors. So this ASXL family of proteins, we have 3 of them in mammalian, and these are the orthologs of ASX in Drosophila. And interestingly, mutations of these ASXL factors are all associated with rare diseases, the Borin-Opitz syndrome, the Shashipina syndrome for ASXL2,
and the Bainbridge-Roper syndrome for ASXL3, indicating their important role in development. So in the Jurgmiller Nature paper, where he described that the ASXL promotes deubiquitination of histone H2A by BAP1, we used that study basically to start mapping which are the motifs responsible for this deubiquitination. And here you can see ASXM, which is also called the deubiquitinated domain in ASXLs. My apologies for that. You will see perhaps both ASXM and the deubiquitinated in the presentation. So basically, we start mapping how the ASXLs interact with BAP1, and in fact, we identified the ASXM, which is also called the deubiquitinated for the adapter, as the domain that is necessary and sufficient for interaction with BAP1. So you can see here clearly with the mapping that the ASXM is responsible for the interaction. And since the same platform in ASXM, the ASXM in the ASXLs is used for interaction, one can predict that they will be mutually exclusive complexes. So basically, BAP1 interacts with ASXL1 or 2 or 3 using this ASXM domain to form mutually exclusive complexes. So then, what is the domain in BAP1 responsible for the interaction? And I will not go into details, but basically we identified the CTD domain of BAP1 as responsible for that interaction. And more importantly, this interaction that involves the ASXM and the CTD is required for promoting the deubiquitinase activity of BAP1. These are transfections in 293 T cells, and this is the non-modified histone H2A. This is the monoubiquitinated histone H2A on lysine 119 for both ASXL1 and ASXL2. You can see that when we remove the ASXM from ASXL1 or from ASXL2, we abolish completely the deubiquitinase activity of BAP1. So again, this has been published, but basically what we have been characterizing using in vitro studies and defining what we term a composite ubiquitin binding interface that involves the UCH domain, the CTD domain, and the ASXM or the deubiquitinated, all of them making contact with ubiquitin. And in fact, we defined, in addition to the hydrophobic patch used for the interaction with UCH, a charged patch of ubiquitin that allowed the interaction with the CTD and ASXM. So this composite ubiquitin binding interface that forms is required for catalysis, and we were happy to see that structural studies done by others have supported this model of composite ubiquitin binding interface promoting catalysis. So things have become a little bit more complicated when we found that basically the deubiquitinated itself, the ASXM, become monoubiquitinated in the presence of BAP1. So this is HEC293T transfection experiment on which we see that the deubiquitinated, the ASXM, become monoubiquitinated. I'm not going to go into details. Here we have the endogenous ubiquitin, the HA transfected ubiquitin. We have also some phosphorylation going on, but this we have been characterizing this very carefully by IPs as well. And we can see that on the full length proteins. So for ASXL1 or ASXL2, in this IP experiment, we can see that they become monoubiquitinated in the presence of BAP1. We characterized the site of monoubiquitination in ASXL1 and ASXL2. It's lysine 370 in ASXL2,
lysine 353 in ASXL1, and this site is highly conserved from human to Drosophila. In fact, if we use Drosophila cells, the ASXM domain, we can find that it is also monoubiquitinated and it's dependent on BAP1. If we do the RNAi for Calypso, we completely abolish this monoubiquitination of this domain. And what is interesting is that this monoubiquitination also requires this proper folding of BAP1, this intramolecular interaction that brings the UCH close to the C-terminal part of the protein. In fact, if we express either the UCH or ASXM, UCH and the CTD with ASXM, or UCH with ASXM without the CC1, we don't see much ubiquitination. Some of it may be because of the endogenous proteins, but more importantly, if we have now the UCH, the CC1, and the CTD expressed separately with the ASXM, we can see again the monoubiquitination. And we can, with the discrete mutation, abolish this in the CTD, abolish this monoubiquitination. Therefore, this proper folding of BAP1 is also very important for having the dubate, the ASXM domain of ASX cells monoubiquitinated. So we wanted to know what is the impact of this monoubiquitination. And to make a long story short, and this has been already published, is that this monoubiquitination can regulate stability. We have this cycloheximide chase. The monoubiquitinated form is more stable, but the mutant is even strongly stable. It's more stable. And we provided evidence that this monoubiquitination site is also the site used for chain extension and degradation. Because when we, for example, inhibit the PSMD14, the ubiquitinase of the proteasome, we can see a chain extension on the dubate. We identified the enzymes that do this monoubiquitination, and I will not go into details. This is with the E2 enzyme. We have this family called UBE2E3 enzyme and E1, so they are responsible for this monoubiquitination. So this monoubiquitination promotes catalysis because when we mutate the sites, we don't see the deubiquitination of histone H2A detected with specific antibody. And in vitro assays with purified components, we can see also that the mutant is inactive or, comparatively to the wild type in deubiquitinating histone H2A. And also in Drosophila, when we express in the imaginal discs of Drosophila the ASX with the mutant in that monoubiquitination site, we can see nicely that when we express the wild type, we see a nice deubiquitination with the antibody for histone H2A. But when we express the mutant, not only we don't see that deubiquitination, but we see an increased signal, which suggests a dominant negative effect. I will not go into details, but basically we have phenotypes in terms of cell proliferation, in terms also of Drosophila development. And importantly, we identified mutations. It's a discrete mutation, only 4 amino acids in the CTD, that basically when we purify the complexes, we can see that we lose BAP1 interactions with the ASX cells, and it's completely inactive in deubiquitinating histone H2A. So just to summarize this part, basically what we believe is that we have a symbiotic relationship between BAP1 and ASX cells. The ASX cells, when synthesized in the cytoplasm, they get monoubiquitinated, eventually polyubiquitinated and degraded. And if they meet BAP1, they get stabilized and monoubiquitinated. And that in turn promotes the deubiquitinase activity of BAP1. On the other hand, here, when we have mutations that they will abolish this interaction, we have basically an ineffective BAP1 in terms of deubiquitination and also in terms of regulating the epigenome. So the last part of my presentation, I know that I have few minutes left. I will try to go quickly on this. So basically, we ask the question how BAP1 is recruited to chromatin, and we have these transcription factors called FOXK1 and FOXK2, which are basically interacting with BAP1 through a phosphorylation site in BAP1. And basically, what we did in these human primary cells, we expressed FOXK1 and FOXK2. And to make a long story short, we can see that FOXK1 promotes cell proliferation, but not FOXK2. FOXK1 prevents senescence in this model of primary cells. And also FOXK1 is a potent oncogene that promotes oncogenic transformation when expressed with different oncogenes. And we can see here the formation of tumors in mice. This is the FOXK1. So this is the property of FOXK1, but not FOXK2. We did the RNA-seq experiment, and then for FOXK1 and also FOXK2, and we can see that FOXK1 specifically promotes the expression of E2F target genes, and here we have the signature of E2F target genes. Now, FOXK1 is known to be amplified and overexpressed in cancer, and this is associated with a poor prognosis. So we separated gene expression data in low and high FOXK1 expression, and we observed that when we have a high FOXK1 expression, this is associated also with the expression of E2F target genes and the signature of E2F target genes. However, when we did ChIP-seq experiments, we were surprised to see that there is almost the same profile of FOXK1 and FOXK2 in terms of recruitment to chromatin, yet we see, again, E2F target genes in the gene ontology. And as you can see here on E2F target genes, you can see the occupancy by FOXK1 and FOXK2. So we wanted to know what's the difference between FOXK1 and FOXK2. They both occupy the genome in the same way, but only FOXK1 promotes oncogenic transformation and promotes E2F target gene expression. So we thought that maybe OGT, which is an enzyme of the complex, maybe can distinguish between this regulation for FOXK1 and FOXK2. And to make a long story short, FOXK1 is in fact gluconaculated by OGT, but not FOXK2. These are in vitro experiments showing also the same thing. And when we do the RNAi for OGT, we can reduce this gluconaculation of FOXK1. We can see that this gluconaculation of FOXK1 increase when we modulate cell metabolism, for example, with glucose. We can see also during cell synchronization experiments that the gluconaculation of FOXK1 increase coincident with the expression of CDC6 in synchronized cells, which is also an E2F target gene. So we did the mapping experiment to see which motifs are gluconaculated by OGT in FOXK1. To make, again, long story short, the C-terminal parts, and we identify by mass spectrometry the sites. We have 11 gluconaculation sites in FOXK1, and when we mutate them, we reduce this gluconaculation of FOXK1. We had to come back to cell proliferation assays, and in fact, when we mutate the sites, we reduce cell proliferation, and we reduce also the tumor promoting ability of FOXK1. Now, when we go again to do the occupancy on the genome, we were surprised to see that we didn't see changes between FOXK1 and the gluconaculation mutants. But we found that the recruitment of BAP1 on promoters is reduced between the FOXK1 and this gluconaculation mutant. And for the gene that they are differentially regulated, we can see again the signature of E2F target genes. I will not describe this knock-in mice, but basically we generated knock-in mice for BAP1 that they are defective interaction with FOXK1, and we have basically similar phenotypes. So to conclude, we believe that the gluconaculation of FOXK1, in addition to the phosphorylation of BAP1 on threonine, stabilize FOXK1 on the chromatin to be able to deubiquitinate histone H2A and promote the expression of the E2F target genes. And that this modification is very sensitive to cell metabolism, because not only it's responsive to cell kinase signaling, but is also responsive to this UDP N-acetylglucosamine, which is derived from basically four metabolic pathways. So basically, this metabolite, by regulating OGT, can modulate the recruitment of BAP1 complex with FOXK1 to chromatin. And with this, I think I end my presentation. I will thank all the students and that have been in my lab contributing and working on this project, and my collaborators, and thank you very much.